Welcome, everybody, to the Major Issues in Modern Biology Lecture Seminar Series. Today, we're lucky to have Dr. Ira Mellman, who comes to us from Yale University, where he is the chair and Sterling Professor of Cell Biology and Immunology. Woohoo! He's also a very prestigious member of the Ludwig Institute for Cancer Research. He holds too many positions and has gotten too many awards to really go through, unless you want me to, for about a half an hour, which I won't. But most importantly, he touches almost all of the cell biologists in some way by being the editor-in-chief of the Journal of Cell Biology, so we should be kissing his butt. He was, yeah, okay. He was recently elected as one of only 3 Americans to the European Molecular Biology Organization, which is quite an honor. He became a faculty member at Yale in 1981. Sorry to date you. After completing his undergraduate degree at a small liberal arts college in Ohio, Oberlin, which is known for its music. But he was seduced by science and actually published his first paper in the Journal of Cell Biology there. He received his PhD at Yale in human genetics and began his postdoctoral work and his academic career at Rockefeller. He's been a leader in understanding the fundamental mechanisms that underlie membrane trafficking events, but he's also been a pioneer, which is something a little bit different, and a real mentor to all the other cell biologists in his ability to apply what he's learned mechanistically towards understanding the higher order roles and physiological functions of these mechanisms that you can define. And these higher roles that he's been studying in terms of membrane trafficking include cell polarity, which we all know his work on, and more recently, the immune response, which is what I think he's going to talk about today. And the title of his talk is A Cell Biologist Views the Immune Response.
That was great. I can take you everywhere. Although I thought I was going to get a kiss. It's a pleasure to be here. I've never been to Davis before. It's quite beautiful, and there's lots of friends here, which is always nice. And these sort of general or special lectures, I always feel I should not just give a regular typical science talk, but rather say something that at least, even if you have no interest or care about anything else that I'm going to talk about, at least you'll connect with something. And so today's diatribe will be really about the immune response or immunology in general. And I find it, as someone who really was brought up in the current generation or previous generation of molecular cell biology, quite always amazed by the fact that immunologists and cell biologists have an almost impossible time talking to each other, despite the fact that they both can really attribute their histories to almost a single event, almost a single experiment that was done by this guy, who is not Peter Walter, or Johannes Brahms, but rather Elie Metchnikoff, who is shown here in his laboratory at the Pasteur Institute in the whatever century. It's not last century, 2 centuries ago, late 1800s, early 1900s. Metchnikoff, here pictured with his microscope, did something that probably many of us in science would like to do, but our families would go nuts if we did. He brought his microscope with him on vacation. And on the beaches of Messina, where the sun was very good, he had a tendency just to do experiments while his family was off playing in the surf. And one of the most famous experiments he did was to take starfish larvae and impale them with various foreign objects, pieces of sand, wood, small stones, bacteria. And what he found was shown here. This is one of his early drawings. That a series of cells would basically come in from this and surround the foreign objects that he put into these larvae. And some of the cells were large, some of the cells were small. The large ones he referred to as large eaters, which in Greek is macrophages. And what these cells would do would be to take up the foreign objects. So here's one particular experiment in which the foreign objects, in this case, were bacteria. And what he did with this was to show that these are bacteria that are not only taken up by the cell, but are actually localized into an acidic environment inside. So he established that endocytosis exists. This is long before anybody knew anything about membranes, that macrophages eat the bacteria, that the bacteria then go to an intracellular acidic site. He knew this because what he did was to take these cells and treat them with a dye called neutral red, which partitions into acidic environments. And he found that all the bacteria turned red, which means that they had encountered a low pH environment. The bacteria then are killed, meaning they cease to grow. And the bacteria then disappear. And they don't disappear because they get expunged or expulsed or exocytosed by the cell, but rather they get degraded in situ. So he basically defined the entirety of the endocytic pathway, that there's uptake of a particle, the particle is delivered to an acidic site. The acidic site is responsible for digestion of the particle, much as you would expect for the gastrointestinal tract. He also found that all of this required that the macrophages be alive. In other words, if you use metabolic poisons, none of this would happen. Or if you poison the cell at various stages after uptake. So if you took a macrophage with the bacterium was red, you then killed the macrophage, the red dye would leak out. So he knew worked basically all of this stuff out. And what this meant was two things. First of all, not only from the point of view of a cell biologist, he was the first real instance of membrane traffic, of a higher order event that a complex cell can do. But from the point of view of immunology, what he realized in this and many, many subsequent experiments is that this was the basis of the entire immune response, or at least one whole arm of it. We'll talk about the second arm in a moment, which was then referred to as cellular immunity, now is more commonly referred to as innate immunity. And what he perceived from these rather simple experiments is that all organisms, all complex organisms, have the capacity to recognize foreign invaders, detect them, and kill them, thereby protecting the host. Okay, so two absolutely most critical observations that you can imagine in modern biomedical science, really attributed to one of the most simple and elegant series of experiments that I think in my observations of science over the years really have been done. And really, you can argue about whether you can come up with this or a better example, but to me this is a node in which one who is a cell biologist can trace your origins back to a very large extent, and if you're an immunologist also would trace your origins back to a very large extent. Yet since that time the two fields have diverged and essentially live in parallel universes, and so I can insult both groups equally, although I suspect there are probably very few immunologists here. I find that immunologists, now that I exist as in this purgatory of being in neither camp and doing this kind of work, immunologists have almost no interest in understanding how the cells that they study work. They're just Lego bricks that provide functions that allow large, complex arrays of immunity to be built up. But they have some level of tolerance for people who are interested in the cell, but really deep down inside they couldn't care less. Cell biologists have the almost opposite pathology, and myself included to a large extent, which is that a cell biologist's interest in a problem decreases by the square of the number of cells involved. And this also is a problem, because I think as we look forward into what really are the major problems in biomedical science, you see I'm trying to fulfill the charge of this lecture series. It's really trying to understand how it is that we can all not only learn how to do reductionist science in the best tradition that has been established over the last 2 or 3 decades of work in modern cell and molecular biology, but how then to apply that type of reductionist logic to ever more complex problems, up to and including human biology and human disease. That doesn't mean that we stop doing one to do the other, because in fact the second cannot be done without the first, which has been the problem all along with doing clinical research. It's not based in the types of foundations that it really needs to be based in. But having said all that, enough of scientific philosophy. Let me just show you where we picked up this problem, just so you can get some idea what brought me to the point I am now, and why you're going to be hearing about this today. This is one of the very first diagrams that I actually, my first wife, who I'm still married to, drew. When my good friend and colleague Ari Hellenius and I started our lab in the early 1980s in New Haven, we really had no more than 2 chairs and a desk and not very much else to do. And so we started working on these structures, which are now very widely understood and distributed in various systems, which we termed endosomes. And obviously, I think probably everybody here now understands in one or another setting that endosomes receive almost all macromolecular content that comes in from outside the cell and really provide a site where receptors and ligands are sorted, where the ligands are, generally speaking, moved to lysosomes where they're degraded. These are the structures that Metchnikoff studied. And the receptors are returned by recycling out to the outside world. So after doing this for a few years, both of us began to get a little bit bored, I should say, with the pathway and started moving in somewhat different directions. The direction I took here from another very early diagram is the problem of membrane traffic in immune cells as it related to the process known as antigen presentation. And what that means is that macromolecular protein antigens, generally speaking, in order to be recognized by T cells in the immune system, this is now the antigen-specific or humoral arm of the immune system, which we'll come back to in just a minute. The antigens themselves as protein entities really are not capable of being recognized, but rather they have to be broken down into short peptides and loaded onto one of 2 types of MHC molecules, major histocompatibility molecules, class 1 or class 2. The class 1 system generally surveys antigens, foreign antigens such as occurs during a virus infection, that are made endogenously by the antigen-presenting cell. They're synthesized in a variety of different intracellular compartments, the cytosol being one of them, where these proteins are ubiquitinated, are digested by the proteasome, and then short peptides are translocated into the endoplasmic reticulum by an ATP-dependent translocator called TAP. Loaded onto class 1 molecules, which essentially completes the folding pathway of these molecules, and they then are transported from the ER through the Golgi directly out to the cell surface. The class 2 pathway, which is what initially interested us predominantly, services foreign antigens that enter cells from the outside, so such as Metchnikoff's bacteria, various protozoan parasites, tumor cells, a variety of other types of pathogens or foreign material. These are taken up by endocytosis, delivered to one or more endosomal or lysosomal compartments where peptides are generated. They encounter newly synthesized MHC class 2 molecules, which on their biosynthetic pathway are diverted from the Golgi complex, and instead of going directly to the surface, instead go to the endosomal compartment for as yet really undetermined reasons, and we'll come back to this in a minute as well. The newly synthesized class 2 molecules are then loaded with endocytically derived peptides, and then these also are recycled out to the cell surface, enabling 2 different classes of T cells to recognize these peptide MHC complexes, so-called CD8 positive T cells, and for class 2, CD4 positive T cells. But we don't really need to worry about that today. Now, so to try and study this process, which in the early 1990s, when we first became interested in it, there were really very few cells that were available in the laboratory that allowed you to really look at this, because it turns out that not all cells are capable of doing this, largely because only a restricted number of cell types, such as macrophages or B cells, really at that time were understood as making MHC class 2 molecules and therefore being able to stimulate immune responses. So our choices were pretty much shown here. All of them were really pretty sorry. B cell lines weren't so bad, which is what we started with. Transfected cells in those days were very difficult to come by. And it was around that time that I got a call from my ex-postdoctoral advisor, who was still and is still at Rockefeller University and is one of my closest friends in science, Ralph Steinman, who had really just over the previous few years been developing the identification of a new cell type called dendritic cells. And what Ralph pretty much said was this, that we should study dendritic cells, quote, why waste your time with anything else? The reasons for that were actually many, because dendritic cells had barely been isolated by that time, or at that time. There were no cell lines, and it required the killing of mice to do this, which was anathema, at least to me, and again, with still small kids running around the house, I didn't want to have to explain, what did you do today, Daddy? Oh, I killed 50 mice. So Ralph, though, is, if nothing, he's many things and probably will win a Nobel Prize for this discovery, but he is nothing if not committed to science and committed to his friends. And so what he decided to do was to send us cells on a regular basis. And this is a picture of the train to New York from the New Haven station. New York is in this direction. Ralph at the time lived in Connecticut, and for a period of about a month, he'd leave work with a box of dendritic cells, get onto the train, drive all the way up to New Haven with it, and hand it off to somebody at the train station. I, of course, was too busy to be bothered with this. So a postdoc or a student would come and meet Ralph, get the box. Ralph would walk across the platform and get the next train back down to Westport, Connecticut, which is where he lived. But even he got sick of this after a while and then decided to simply leave the dendritic cells inside the train, which worked for a while. But again, this is all before 9/11. And one day, one of the postdocs went down and said that she couldn't find the cells, they were gone, and we decided that at that point we had to just simply do it, and so we did. Now actually there's an interesting scientific sidelight, which if you were immunologist I would go into as to there was something actually very special about this trip up, because this train is so bad and shakes all the time that the cells are actually changing because they were being shaken for an hour and a half relative to what the cells looked like when we got them. And actually, I'll show you what the difference is. And it really was one of the more astounding observations that we made in the system, which is really what got us so excited about them. So why all of this trouble and endless, at this point, at least it may so seem, introduction? Now, dendritic cells are by far the most efficient of all antigen-presenting cells. They can stimulate immune responses with almost non-existent amount of antigens. B cells also do a very good job, for those of you who know about B cells. But B cells, because they capture antigen, because they have a specific antibody molecule they make as a membrane protein that's specific only for one and only one antigen, they're really good, but only can do it in one setting. Dendritic cells basically are as good for almost any antigen that you can throw at them. They have the capacity of being able to take antigen from the periphery to lymph nodes. And what's meant by that is that they exist in almost every peripheral tissue, skin, the gut, lung, brain, wherever you like. And they wait there as sentinels until a foreign antigen arrives, receive it, interpret that as a signal, and then migrate through the lymphatics to lymphoid organs where they find T cells. They have all the molecules that they need. They can stimulate a wide variety of different T cells, but most importantly, or different immune cells, but most importantly, they stimulate what one called immunologically naive T cells. So these are T cells that have a defined specificity, but have never before seen it. Now, to get an immune response going, an antigen-presenting cell must exist that will educate a naive T cell to understand what it is it's supposed to be seeing, so that it can either kill that or assist B cells to make antibody molecules to that cognate antigen. So what that means then is that without dendritic cells, there would be no initiation of any antigen-specific immune response. In fact, one can show this genetically by knocking out various genes that are required for the development of dendritic cells and showing that these mice effectively become totally immunocompromised. But kind of almost as a Faustian bargain associated with forming a cell that is so good at generating immunity, these cells also have a second and equal function, which we won't have time to talk about today, but I did want to at least bring it up, which is the maintenance of what's called peripheral tolerance. This is an absolutely key feature of the immune system, and I think probably represents, from a systems point of view, the most important problem that faces us as scientists. I won't say immunologists because it faces everyone, which is how is it that the immune system on a minute-by-minute basis can distinguish between self and non-self? How does the immune system know that such and such is a foreign antigen and therefore should be recognized as such and used as a target to generate an immune response versus a endogenously expressed protein or piece of DNA or anything that is associated with the normal steady state situation? There's some ideas as to how this occurs. Our view is that dendritic cells play a key role in this. Now, I may mention this briefly again, but we really don't have a good solution to this problem yet. Now, what we do have a much better solution to, and we're really, I think, the cell biological features of this system really become quite compelling, is that virtually all of these features are regulated by a process of terminal differentiation, which we refer to as maturation. Now, what is dendritic cell maturation? This is, again, a slide or a definition that is as appropriate to immunologists as non-immunologists because it's a very poorly understood process in terms of its function. So I define it as any stimulated process of terminal differentiation that enables dendritic cells to facilitate various immune responses, immunity or tolerance, meaning anything. So it basically, the idea is it turns them from a cell that basically does nothing except capture antigen to a cell that can now regulate T cell responses. The way this works from an overall systems organization point of view is diagrammed here. So here are sentinel dendritic cells that are present in a peripheral organ, in this case the skin, where they're referred to as Langerhans cells. Yes, it's the same Paul Langerhans who discovered these cells as discovered the islets of Langerhans in the pancreas. So these cells exist in the epidermis or the dermis. They wait for antigens to come, bacteria, viruses, whatever, and then they leave the periphery, enter the afferent lymphatics, and then find their way to lymph nodes and enter then the so-called T cell zones. These cells in the periphery we refer to as being immature because if you isolate these cells and look to see what they can do, they can take up antigen very well, but they don't make peptide MHC complexes very well, nor as a result do they stimulate T cells very well. On the other hand, dendritic cells that you isolate from lymphoid organs, in fact, do all of those things. They may not be able to take up antigen so well, but what they can do is make lots of peptide MHC, lots of hormones or cytokines or other molecules that are needed for T cell stimulation, and they're just absolutely wonderful at that. Whereas, as I said, the immature cells don't. So there's a maturation event that converts them from one function to another, and that corresponds to a period of time and also obviously a different location in the cell. Now, it's not due to the location that they change their activity so much, but rather due to a rather dramatic cell biological transformation. But just to make one further point on that and why this is so important as just a basic conceptual insight, which is dendritic cell maturation because of what it is. In other words, it's something that is triggered by the detection of a microbe. Basically connects both arms of the immune response. So it connects innate immunity, which, as I've already mentioned to you, was really first described by Metchnikoff, which really comprises a direct cellular response to pathogens based on the recognition of shared patterns, if you will, or shared elements, lipopolysaccharide, methylated DNA, double-stranded RNA in the case of viruses. There are a series of receptors that dendritic cells have that will detect all of these chemical entities, and all of these chemical entities will trigger maturation. So it detects these activities to the adaptive immune response or the antigen-specific immune response, discovered and described really pretty much at the same time by Paul Ehrlich, who you may know better because there was actually a movie made about Paul Ehrlich called The Magic Bullet, in which Paul Ehrlich was played by Edward G. Robinson. Maybe we can get Peter Walter to play Metchnikoff in a movie. But what the adaptive immune response does is this reflects the ability of T cells to guide the production of antigen-specific killing mechanisms and also guide the production of antibody molecules, which we all know and love as being responsible for protecting us against a wide variety of pathogens. Now, all of this is well and good, but what really struck our group, I think, as cell biologists about the process is that if you look at mature and immature dendritic cells, they look like this. So immature dendritic cells have this appearance, mature dendritic cells have this appearance. What you're looking at is just a conventional immunofluorescence picture of cells stained for MHC class II molecules in green and a lysosomal protein in red. And what you can appreciate already is that the immature cell has most of its MHC molecules present in the lysosomal compartment, whereas the mature cell has most of its MHC class II molecules present on the surface, and now the surface has extended these long dendrites and membrane folds that basically give these cells their name. So these are the cells that you find in lymph nodes. These are the cells that you find in peripheral tissues. And you can generate a mature cell from an immature cell just by treating the cells with bacteria and waiting for something like 10 to 12 hours. So it's a remarkably rapid change that involves massive alterations in gene transcription, but also massive, not only post-transcriptional, but post-translational changes, as I'll show you in a minute, that enable this rather remarkable transformation of a cell from one to the next. And what makes it special is not only because there's just so many neat things that are going on that affect membrane traffic and cytoskeletal organization, but also, as I said, there's a real systems context to this, which is really what I'm trying to stress. These cells have two completely different functions, and by learning about how you convert a cell from an immature to a mature dendritic cell by studying the reductionist aspects of the cell biology that are really responsible for this transformation, you can ask one after the next what each one really means when you put it in the context of understanding how the immune response works. Now, a great deal of work was invested over the last several years in trying to understand just the alterations in membrane traffic that occurred. And I'm not going to, since most of this has been published, at least what I'll summarize here, I'm not going to go through the primary data, but just rather use this articulated diagram to show basically what happens in a summary form. So on the left is basically the membrane traffic phenotype, if you will, of an immature dendritic cell. So here are cells that take up lots of antigen from the outside by phagocytosis, by macropinocytosis, or by clathrin-coated pit-mediated uptake. Macropinocytosis and phagocytosis being rather similar processes depending on active Rho family GTPases such as CDC42. MHC class II molecules are made and are targeted to the lysosomal compartment because at this point in their lives they're associated rather stably with a chaperone called invariant chain, which is thought to have lysosomal targeting elements on it that divert these molecules from the Golgi to the endocytic pathway, enabling them to be sequestered in the endocytic pathway. So what happens shortly after these cells receive a maturation stimulus is that levels of active CDC42 in the periphery fall. So instead of having a lot of GTP-loaded CDC42, you now find there's a precipitous decrease, and now most of the CDC42 in the cell is present in the inactive GDP-bound form. That has the consequence of basically shutting off two of the major forms of endocytosis, phagocytosis and macropinocytosis. So that explains one major change. Newly synthesized MHC class II molecules now proceed to the surface. One of the major reasons for that is because an antiprotease called cystatin C is downregulated. Both it becomes a secreted protein and also its transcription is turned off. What cystatin C is a very potent inhibitor of a cathepsin called cathepsin S, who has as its main role in life cleaving this invariant chain. So the reason the invariant chain is very poorly cleaved in the immature cell is because there's very low cathepsin S activity due to the high amount of cystatin C. When the cystatin C is downregulated, cathepsin S activity goes up, although the amount of cathepsin S doesn't change at all. And now invariant chain is cleaved very rapidly, and these newly synthesized class II molecules, although they're still diverted to the endocytic pathway, now instead of going to lysosomes, go out to the cell surface. Next, antigens that had been taken up by the cell during or prior to the maturation event are now pressed into service. So the antigens which had previously accumulated in lysosomes in a non-degraded fashion are now very rapidly degraded and loaded as peptides onto the MHC class II molecules. Now the reason this happens is also incredibly neat, because in the immature cell, the lysosomes have a relatively neutral pH, at least by the scale of lysosomes. They have a pH of about 5.5. But it turns out that most lysosomal proteases have a rather sharp cutoff for degradation of protein substrates. So they're almost completely inactive at 5.5. I shouldn't say that. They're about 5% is active. 2 to 5% is active at pH 5.5 as they are at pH 4.5. So as soon as you mature the cell, the pH drops. And it drops because you actually assemble the vacuolar ATPase, which is an F1 FO-like enzyme that we found years ago on the surface of endosomes, that when it's assembled from the two major complexes that form the ATPase, these vacuoles, the lysosomal vacuoles, become more acidic, and the proteases become activated. The antigens get degraded, loaded onto class II, and the class II molecules then go out to the cell surface as a consequence of formation of a rather remarkable array of tubules that emanate out of the lysosomal compartment and eventually fuse with the plasma membrane. This is just some old pictures taken by a graduate student, Amy Chow, who figured out how to introduce a GFP class II molecule into dendritic cells. This has now been done, I'll show you in a minute, more elegantly by Hida Plug, who made a mouse. But the retroviral system that Amy has used is really kind of useful for other reasons. So this is an immature dendritic cell with labeled lysosomes that are just sort of bouncing around. If you take these cells and now treat them with an activator of dendritic cell maturation, you can now see that tubules start shooting out of these structures. And if you wait long enough, you can start seeing the class II molecules appearing on the cell surface. Now, the lysosomes themselves are really rather interesting, because if you look at them, they have a rather characteristic abundance of these internal vesicles. And to people who study the endocytic pathway, these are what we call multivesicular bodies, because there are bodies or lysosomes or late endosomes that have lots of little vesicles inside of them. And what's always been rather confusing is the fact that most of the MHC class II molecules that are found in these structures are not on the limiting membrane, but rather are on the internal membranes of these structures. Which is kind of confusing, because most of the time when you're looking at receptors that are downregulated as a consequence of endocytosis, such as epidermal growth factor receptor or insulin receptor, they're sequestered on these molecules and thereby marked for degradation. In the case of class II, however, they seem to be rescued, and how they're rescued is completely unclear. Nor is it clear how they get in there in the first place. But in any event, one of the ideas is that the vesicles, as a consequence of being triggered, as a result of triggering the maturation process, may do a fusion event that hasn't been seen before, which is an ectoplasmic fusion of the vesicles with the luminal surface of these structures, introducing the class II molecules, providing the extra membrane, if you will, that allows these tubules to be formed. Now, this is just another set of pictures. These are taken actually with the GFP class II mice that we've now also put a lysosomal marker in, just to show in slow motion that these tubules can really be quite long. We can show that eventually they do fuse with the cell surface using biophysical techniques. But just want to make the point that what's really remarkable about them is that the lysosomal resonant proteins, such as here is LAMP RFP protein, extend out in the tubules together with the class II molecules. Now, but if you look at what happens at the cell surface, only the class II accumulates. The lysosomal membrane proteins don't. So somewhere along the line, there's a sorting event taking place. And we think that the sorting event takes place actually directly at the cell surface at the time of fusion. So here what you're looking at is a two-color total internal reflectance image of a cell in which there's a hotspot for fusion that occurs right here. And what you can see is a continuous appearance of vesicles that are green, then they're yellow, then they're red. Or they're yellow, then they become green, and then they become red. So green rather is class II. red are the lysosomal proteins. And I'll show you stills of this, which may be a little bit easier to see, but I just want you to appreciate the fact that there's an enormous amount of dynamic that occurs in this region where there's green and there's red. You really can't figure out very much what's going on, but nevertheless, there does seem to be at least color sorting for whatever that's worth. So here, what you're looking at now is simply a series of frames of just the lysosomal protein taken, again, in a two-color image, but here using total internal reflectance in green and conventional epifluorescence in red. So what this means is that first you see a red tubule approaching. The tubule then turns green as it either touches or fuses with the membrane, because this is how, I won't describe how the technique works, but it's a way of measuring events that occur either at very, very close proximity to membranes or that are representative of fusion events. So it becomes very green, which means it's either fused with or very close to the membrane, and then it pulls back and becomes red again. So what we think this may mean is that you get a transient fusion event and sorting occurs at the cell surface. And in fact, if you do a series of FACS experiments measuring the appearance of these LAMP molecules on the surface, they come out, and then over the next period of time, they seem to go back. And so the general idea then is this, is you get this mixed population tubule coming out, and then the LGP or LAMP molecules, presumably because they still have a clathrin-coated pit localization signal, can very rapidly retrieved and may in fact be retrieved at a site that's very close to where the fusion site takes place. So this is obviously ongoing work, and we haven't quite figured out precisely what the mechanism is. But I'd like to move to something that I think is even more intriguing about this, which is why this occurs at all. I've already told you that endocytosis is supposed to stop during maturation, but it doesn't really. Here, I guess I already said that what occurs is both phagocytosis and macropinocytosis are the two major forms of endocytosis in the immature cell. These are what gets turned off as a consequence of maturation, but it turns out that the uptake of plasma membrane proteins via receptor-mediated endocytosis by clathrin-coated pits and coated vesicles seems to continue unabated. So all sorts of things get taken up. The question is, why don't the class II molecules get taken up? Because it has long been presumed that they contain some type of clathrin-coated pit localization signal. So that brings us to trying to understand how that might work. And the problem is that there is not really a good clathrin-coated pit localization motif on class II molecules, which led us then to wonder whether or not there might be a post-translational modification such as ubiquitination, which could play a role. And this is a diagram taken from a paper, a review of Harold Stenmark, showing just basically what's thought to happen in the case of epidermal growth factor receptor, which when activated as a consequence of ligand binding will cause a ubiquitin ligase, in this case Sybil, to add what is often thought to be a monoubiquitin, but probably is more than simply one, to one or more sites on the EGF receptor cytoplasmic tail, which interacts with the various number of ubiquitin-specific adapters that sequester these proteins, the EGF receptor in clathrin-coated pits, and take them to endosomes, where something else happens that's really rather quite interesting, which I've already mentioned actually. The ubiquitin is then recognized by a series of 3 or 4 different protein complexes that sequester these receptors for yet another type of vesicle formation event, this one associated with the formation of these internal vesicular structures that I mentioned as being involved in downregulating these types of receptor tyrosine kinases in MVBs or lysosomes, but that also seem rather provocatively to contain class II molecules, which we don't think are downregulated. In fact, we think they're rescued and taken back out to the cell surface by some mechanism. So Jansuk Shin and Mia Yan got very interested in trying to understand whether or not could any aspect of this system actually play a role in understanding class II transport in dendritic cells during their maturation or development process. So we really have to start by realizing that class II transport in dendritic cells is regulated at least 3 sites. So it's regulated at the level of exit from the Golgi, where rather than going to the surface, it goes to endosomes. It's regulated at the level of endosomes because it is taken in to the MVBs, as I already showed you in the EMs. And it's also regulated at the cell surface because, as I've already shown you again, in the mature dendritic cell, the class II molecule is not internalized. So how can you explain all of these various events? But again, ubiquitin seems to be implicated. This is just a picture of ubiquitin. It's a small 76 amino acid peptide that obviously is very well studied in a variety of settings. So what Jean-Suk realized is that indeed, I don't know why we never realized this before, but there is in fact one conserved residue, a lysine, which is a potential ubiquitin acceptor site that's present on the beta chain of the normally dimeric MHC class II molecule in virtually every species that we've looked at. Every gene, every allele, every species seems to have a lysine at position 4 in the cytoplasmic tail. No other lysines are conserved either in invariant chain or on the alpha chain of the class II molecule. So obviously she wondered whether or not these molecules might be ubiquitinated, and rather remarkably they are. So if you take MHC class II molecules from bone marrow derived dendritic cells that you differentiate in vitro, or splenic dendritic cells that you isolate from spleens of mice, IP them and then blot them with an antibody to ubiquitin, what you find indeed that you get a typical ladder of ubiquitination where you get 1, 2, 3, 4, or 5 ubiquitins added, the average number being somewhere between 2 or 3. So this is what we would consider an oligo-ubiquitination, as opposed to a polyubiquitination, which is what you see if you simply blot the lysate. So most cytosolic proteins that are targeted for degradation by the proteasome get polyubiquitinated in this way. Now we know this is class II because if you then do this experiment on a mouse that has a GFP fused to the beta chain, you can see that this ladder shifts up as you would expect by the predicted molecular weight of the GFP molecule. Now, if you take out that lysine by mutagenesis and then express the mutant class II molecule in dendritic cells, and just as an aside, I just want to point out these are incredibly difficult experiments because we're not dealing with cell lines, we're dealing with cells that will do anything except be transfected or infected by a virus. So what we have to do is we take a mouse from which we've knocked out the beta chain, isolate dendritic cells from those mice, make recombinant retroviruses of the various mutant forms that you've seen, and then come back into the mouse and then try and do biochemistry after use a transfection, a low yield, low efficiency transfection on about 5 to 10% of the cells in the population. So please don't complain about the fact that these bands are not very dark. But they're definitely there. So the idea here is that when you do this experiment, reconstitute with a virus containing a wild-type beta chain or the lysine to arginine mutant, you can see that 1-point mutation prevents the addition of the ubiquitin. Now, most amazingly, you find that there's a dramatic relocalization of where the class II molecules are. So here, if you in the wild type, these are immature dendritic cells, just a single confocal slice. The transfected wild type is in lysosomes, exactly where it should be, but the transfected lysine to arginine mutant is now predominantly on the cell surface. A little bit is found in the lysosomal compartment, but you can see just by comparing these images, they're really quite different. This just shows that you can pretend to be immunologists and do FACS assays, which are actually quite useful because you can quantify all of these things quite nicely. So here, the lysine mutant, and this is the amount of, in immature dendritic cells, the amount of class II that's on the surface. Now, what doesn't happen, though, is that you don't see an increase in the amount of the chaperone invariant chain on the surface. It stays in the endoplasmic reticulum where it belongs. So here is a FACS for invariant chain associated molecules on the surface, and you don't see them. Now, the reason that's important is because the invariant chain, remember, is removed at the level of endosomes. And so what that has told us, albeit a little bit indirectly, is that the invariant chain class II complex is assembled. In fact, we can measure that biochemically, and then it's taken apart prior to the delivery of class II molecules on the cell surface. And this is in the absence of ubiquitination of the beta chain. So what that strongly suggests then is that you don't need ubiquitination at the level of the trans-Golgi network to target these complexes into the endocytic compartment. And in fact, there are many examples of ubiquitin actually doing just that. Now, do you need the ubiquitin to get into the luminal vesicles in the MVBs in these immature dendritic cells? Here, the result is a bit more ambiguous, and all I can tell you is that you do, but not completely. So if you quantify the amount of M8C molecules in internal membranes in cells expressing wild-type class II or the lysine mutant and look at the numbers at the bottom, you can see that depending on where you set your bar, about 70% of the wild-type molecules are in the internal membranes and about 40% of the mutant molecules are inside. So the best I can say is that it's involved but not completely, which may mean something that's of equivalent interest and importance, or perhaps even more interest and importance, which is that M8C molecules may be an example of something that's long been suspected but never really been shown effectively as a ubiquitin-independent cargo for forming these MVBs. Now, the reason that may be of potential interest is because, as I've mentioned, almost all of the other ubiquitin-dependent cargo that we know, such as EGF receptor, is marked for degradation as a consequence of being taken into the MVBs. So perhaps there's two routes in. So there's a route that allows for escape and then a route that doesn't allow for escape. So again, this is another piece of information that needs to be followed up. But the one thing I can tell you that is definitely so is that you need the ubiquitin for endocytosis. So here is a relatively simple experiment in which you express class II molecules, in this case wild type, in this case the mutant, in dendritic cells, and then ask, these are even immature dendritic cells, ask whether or not antibody to class II can be taken up. And here you can see the red antibody being taken up very nicely by the wild type, and not at all, at least at this level of resolution, by the mutant. So in the absence of the addition of ubiquitin to the beta chain of class II molecules, basically nothing gets taken up. That still doesn't explain then why is it that the M8C molecules don't get internalized after maturation takes place. So this is, Jean-Suk and Mia have then extended this to try and figure that one out. And rather remarkably, what they find is that the entire procedure, the entire event of ubiquitin addition is in fact one of the things that's dramatically regulated as a consequence of changing the differentiation state of the cell. So that although both immature and mature dendritic cells make the same amount of ubiquitinated proteins, these are the a Western blot of total proteins from a lysate. If you IP the class II molecules, what you find is, again, as you expect, the same ladder that you've been looking at all along for the wild type class II molecules, but when you do the same experiment on wild type class II expressing mature dendritic cells, now there is no ubiquitin added. So we think we know now what the ligase is that's responsible for this, and it too seems to be turned off, and our guess is that by downregulating the ligase, you downregulate the amount of class II that is accessible to ubiquitination, and then possibly, therefore, stranded at the cell surface after maturation. Now, the way Jean-Suk has tested this is by covalently adding a monoubiquitin to the tail of the lysine mutant, shown here, and asking what the phenotype is of cells that are expressing this mutant. And that's shown here. In the case of immature or mature dendritic cells, immature shown on the top. So here is a cell expressing wild-type class II. In immature cells, it's in lysosomes, as I've already shown you, expressing the lysine to arginine mutant. It's now on the cell surface. And now a mutant that has a covalent fusion of the ubiquitin molecule. Now, despite the fact that these are immature dendritic cells, you now find the class II molecules inside the cell. Here, after LPS addition, these are mature cells. Wild type is on the surface, the mutant is on the surface, but now, despite the fact that these are fully mature DCs, the ubiquitin fusion protein is still taken inside the cell and endocytosed quite well and delivered to lysosomes. And in fact, again, using the same type of antibody assay, you can show that indeed these are molecules derived from the cell surface because antibody can be taken up from the outside and delivered to intracellular compartments. So just to summarize this, and I think probably what I'll do is just rather than address the next topic, just stop there and not try and confuse you too much, but just summarize where these results have told us so far is that in addition to all of the other things that these cells do, every time you look at another basic cell biological principle and ask how does it contribute to the morphogenesis and functional transformation of these cells, we find another one. So here, what we find is that class 2 molecules are ubiquitinated. Precisely where that occurs, we don't know. My suspicion is that it occurs after surface arrival at the plasma membrane, but we don't quite know that yet. And that we find that by regulating the addition of ubiquitin, we can explain why it is that class 2 molecules, after reaching the cell surface in a mature dendritic cell, remain at the surface, but then are in immature dendritic cells internalized. Rather surprisingly, it's not due to the rather large amount of macropinocytosis that these cells do, but rather uptake it probably occurs almost uniquely through clathrin-coated pits in a ubiquitin-dependent fashion. And then finally, there's, I think, this remaining issue of whether or not the formation of multivesicular bodies and the sequestration of MHC molecules as cargo into the MVBs indeed is or is not ubiquitin dependent. We're actually working at the moment with Wes Sundquist at Salt Lake City to try and determine whether or not this will be really the first example of a membrane protein that would represent a ubiquitin independent entry route into the MVBs. And I'm very excited about that possibility simply because it seems so likely that these molecules are actually not degraded but rather preserved. Yeah, so that's, I think probably what I'll do is just stop there. And I've tried to mention pretty much all the names of the people who were involved in this work, which hasn't been published yet. The major players in a lot of the initial work have been, I can show you their names since they're rather important, are Sergio Trombetta, Lilia Delamar. Sergio is about to leave to take a position at NYU Medical School, together with Philippe Pierre, who's not here, who's in Marseille, and Shannon Turley, who's a graduate student, now has her own group at the Dana-Farber in Boston. So again, all of these people, almost all of them, no one is an immunologist here, with the exception of maybe one or two, but embraced a problem in a completely dissimilar system, not so much, I think, because they had an overriding interest in immunology, but rather because I think it afforded them a rather unique, it's not a unique, but it was an early opportunity to learn how it is understanding and interrogating fundamental principles of cell biology that have been worked out in all sorts of model systems. Beautiful work in yeast, in tissue culture cells, in flies and C. elegans, how you can take these fundamental principles and then apply them to higher order problems that then in turn allow you to get a greater understanding of an even higher level problem, in which case, in this case, the progress and cellular basis of the immune response. So I'll be happy to take any questions if there are any.
So is it possible that these class 2 molecules are actually playing housekeeping roles in multivesicular body formation in these cells? In other words, helping other types of cargo get into these preserved compartments, like a housekeeping function almost?
Yes. But it's interesting, it's probably not the alpha and the beta chain, but rather the invariant chain. Because we find if we look at mice that have invariant chain knockouts, what we find is that there are very few MVBs. that are there. All of the right components are there. And the rate of turnover of endogenous lysosomal proteins is actually much higher. The reason for that is probably because there's a rather serious anti-protease domain in the luminal region of invariant chain. So it probably helps downregulate the overall amount of proteolytic activity that's present in these structures. And my guess is that invariant chain doesn't get there on its own, but gets there in association with class 2. So whoever class 2 is getting in, it's chaperoning the chaperone in, which then has another function after it arrives at its final destination.
Any other questions?
Yep.
So I don't know if you said it, but if you drive the variant chain back to the surface, with these mutants, does it actually present antigen?
That's a really interesting question. It does. If you don't get, if you don't put the ubiquitin on, it's actually a difficult experiment to do, again, because of having to reconstruct. We're actually trying to make knock-in mice that have these things so we can do some better biochemistry and functional experiments. But you're limited in terms of the numbers of things that you can actually measure. The initial findings are that indeed you can still load peptide, probably simply on the way out, because there is still this stopover which causes about a 1 or 2 hour delay in transport out to the cell surface. And that seems to be, at least in these totally contrived artificial laboratory antigen presentation assays, enough to get antigen on. Plus, you wind up with cells that have about 50 times more class 2 on the surface. So probably those, the inefficiency of the loading is compensated for by the fact that you get more MHC out there. But actually, it's a very interesting and important point.
So is the acidification of that compartment altered? Very inefficient, compensating.
Yeah. No, it's not altered, but I'd want to. The problem with these systems is, as I said, they're really contrived. They have been selected by the evolution of immunologists for years to always work, which means they select all of the model antigens that are used are the ones that always give you a result. I mean, you look at them and they turn into peptides, pretty much. So what you really need to do is challenge the system in a more physiologically relevant setting, and that doesn't always even mean laboratory mice, because the number of knockouts that people make, our immunology colleagues make, and find no phenotype, and then get depressed and do something else. Actually, there are an increasing number of examples where those mutations then turn up as being incredibly important in feral mice. There's a couple of examples. In Australia, they have these, what can best be described as mouse blooms, where every few years or so you just get millions of mice that just pile up and die. And the reason that they don't take over is because they become very susceptible to an infection, because they're actually lacking a gene for a particular receptor, which in the laboratory gives no phenotype at all. But you can show genetically that it controls populations of hundreds of millions of mice in a natural setting. Mm-hmm.
Can you say anything about the degree of ubiquitination in terms of stoichiometry? I mean, is it all or nothing, or is there some stoichiometric difference?
In general, the stoichiometry is low, but that's the truth of the matter. But the way that's always rationalized is that it's also it's transient, it's dynamic, so that generally speaking, when you are a protein that's being sequestered into a multivesicular body, before you get in, if in fact you had ubiquitin at the time you got in, ubiquitin is hydrolyzed off by a deubiquitinating enzyme, of which there are many. The one that probably does this is in yeast. It's called DOA1 that does this. And the presumptive reason for that is because if there's a limited amount of ubiquitin monomers in the cell, and if you use them all just once, to get into multivesicular bodies, the ubiquitins get degraded as well. So this seems to be a way to recycle them. So at any one time, at steady state, in a system like this, I'd say probably no more than 10% or 15% of the molecules actually are ubiquitinated. It's very difficult to do the kinetics for obvious reasons, but because there is a one-to-one stoichiometry, at least for things like EGF receptor, between getting into a multivesicular body and having ubiquitin, the presumption is that indeed it's a kinetic effect. Mm-hmm.
What can you tell us about the mechanism of the downregulation of the ubiquitination? You mentioned the ligase.
Yeah.
I think you have. Is that the point?
Well, we're screening now to see which ligase it is. There's actually an ex-student of mine who's at Scripps who has all ubiquitin ligases known to man. So we're trying to figure out which one it is. But there's one, NED4, which is just by accident, and it would be a good candidate for doing something like this. Turns out there's lots of it in immature dendritic cells and none of it, so far as we can tell, or at least much less, that you can at least detect by Western blot in the mature cells. So that, of course, just pushes the problem back one step. What's regulating that? So you then have to solve that one. But my guess is that it's probably that. But there is also one other really interesting effect that takes place, which I don't have an explanation for yet, because we don't really know what the protein does. But there's this whole series of ubiquitin-like proteins, such as SUMO or ISG15. So it turns out that in dendritic cells, in immature dendritic cells, there's like no ISG-related proteins. In mature dendritic cells, there's a zillion of them. And whether or not M8C molecules are one of those, or whether or not that is what's regulating the ubiquitin ligase, I don't know, but we're trying to sort that out as well.
Could I have your thoughts on cell death and physiological turnover of dendritic cells in lymphoid tissue? Because there's an inherent puzzle for immunologists in that there are two types of antigen-presenting cells. Those that present to B cells, which are have this funny name of follicular dendritic cells. And they're long-lived cells, they never turn over. And then you have those that present to T cells, like the APCs, and they have a fast turnover.
Yeah.
And I think that's an interesting dichotomy between two cells that are doing exactly the same thing. So what are your thoughts on that? Cell death.
I mean, the problem is worse than that, because they're probably, I'd say, at best count, maybe a dozen different dendritic cell types at this point. The follicular dendritic cells are interesting because they always have been viewed as a stable cell type that actually presents intact antigen on the surface. But it's actually now starting to emerge that even myeloid-derived dendritic cells can do this, because I didn't get into it, but they have a very low protease content, and a lot of antigen actually survives for quite a long period of time. And you can actually use other dendritic cell types, myeloids or so-called plasmacytoid dendritic cells, which are probably more related to the follicular ones, as being direct stimulators of B cells. In terms of turnover, you're absolutely right that the follicular DCs last a longer period of time. Why that's the case, I don't know, but my guess is that it may have to do with this maturation process. If they don't exhibit the same type of developmental effect as a consequence of receiving LPS or other toll-like receptor ligands, then they would have a tendency of being long-lived, because the first step, maturation is effectively the first step to apoptosis, because you turn on an apoptotic pathway, and what keeps the dendritic cells from dying from maturation is engaging a T cell productively, because then the T cell will secrete anti-apoptotic factors as long as it's being fed an antigen or peptide M8C complexes that it likes. But if that's not happening, or if a dendritic cell can't find a T cell that it can stimulate, it dies. It dies, and it's rather unceremoniously eaten by its neighbors. And after that happens, actually everything that was in that dendritic cell can get represented to the next T cells along the line. So it's a horribly complex web of events that take place. I can imagine ways of trying to sort it out, but at the moment we or no one else, I think, really have good data to give any more sense to it than that.
So you're really, it's interesting. So you're saying that if they haven't captured antigen and aren't in the business of presenting it, they're useless. They might as well die.
Yep, and they do. Otherwise, because
That means that the DCs that have picked it up and are presenting it may be long-lived DCs.
they could, but
They never go anywhere because they never come out of it.
they never come out. No, and the lymph node would fill up very rapidly with dendritic cells if that was the case. Because what I didn't mention is that there is a continuous turnover of dendritic cells that occur, or continuous traffic of dendritic cells, I should say, from the periphery into lymph nodes, even in the absence, as you probably know, of any inflammatory or microbial stimulus. And what's thought under these conditions is that these are cells that are migrating under the steady state. They mature, but mature to a form where instead of causing T cells to initiate immune responses, they actually cause the T cells to apoptose, and then they die themselves. And this is at least what I think is the basis for how tolerance works. So if you generate a dendritic cell that moves to the lymph node in the absence of seeing a microbial stimulus, it becomes mature, but it becomes mature in a rather special way. And what that special way is it does not secrete cytokines that T cells need to actually develop. So they get an initial stimulation, and that effectively is their death signal. They divide a few times and then they die. So it kind of works both ways. You can get a kiss of death if you're the wrong type of dendritic cell and you're a T cell that happens to encounter it. Or if you're a dendritic cell and come in and no one asks you to dance because you're not holding anything that anybody is really interested in, then you die. It's a rather cruel system. And then you get eaten by the queen of the prom. So.
On that note, can we thank Dr. Mellman again?
I'm glad I asked.